Okay, so now we have this PRS 415. All right, PRS 415. Just a second. Rodena, the question I will answer you just a second. So let's start it now. So PRS 415. Okay, we click this. Then first of all, you look at the gene. All right. So this is, you see, this is the did you see the arrow? It's opposite direction from the PRS 413. The PRS 413, the promoter is in here, and then the open reading frame, and they express this direction. And for the PRS 415, it's opposite direction. Okay, so that means when you design a forward primer and reverse primer, you have to be very careful, right? So first of all, you look at the number for the promoter. All right, it's a 1770. All right, and then to 2174. So that means the end of the promoter would be 2174. All right, and then the end of open reading frame is 663. All right, 663. All right, 663. Let me just, okay, the end of the open reading frame is 663. All right, and then the beginning of a promoter is the beginning of promoter would be 2174. 2174. All right, so we are going to amplify the region in that will cover 2174 all the way to the 663. All right, and then so we take 250 base pair downstream of the 2174. So 2174 plus 250, that'll be 2424. All right, and then downstream, so 663, you have to extend the 250. So 663 extend the 250, so you minus 250. All right, so that gives you 413. So that you want to do 2424 all the way to 413. So 2424, then you take 20 nucleotide in this region. So we would take the 2424, all right, to 2405. All right, 2424 to 2405, and then 413 for 20 nucleotide. So that'll be 413 to 432. All right, that's 20 nucleotide. So we just write down this number. That's the target sequence we want to get. All right, so 2424 to 2405, and then 413 to 432. So first of all, we identify this region we want, right? And later on we can download the sequence. But you write down this number first, then we go to the download. Okay, download the gene. So I download the gene, and then VMA3. All right, VMA3 download. Right, VMA3, which is simple and straightforward, because we know we will focus on around 600, and also the another 400 downstream of the stop codon. Right, so we download the sequence. All right, and then after we download the sequence, okay, so now we can open the file. Right, so I'm going to open the file. Open the file, serial cloner. Serial cloner, and then, all right, so now first of all, let's open up the PRS 415. PRS 415 open. All right. So we have PRS 415 available now. And then we open the VMA3. So now we have 2 files available, okay? And now there's a very tricky part, all right? For the VMA3, for the PRS 415. So when you look at the map again, this is opposite direction against the sequence orientation, all right? So that means if the sequence you take from here really backward, then you have to do the anti-parallel of the sequence here. So that means at this time you are going to design a forward primer from here, reverse primer from this end, and so they amplify this direction and this direction. And then the sequence you see, all right? So the sequence you see here, all right, it's the top strand, which is 5' to 3', this orientation. But the LU2, which is a selectable marker that we're going to replace the VMA3, it's opposite direction against the sequence orientation. So therefore, the sequence that we took from here, you have to do the anti-parallel. And each sequence you took from here, you do not need to do the anti-parallel. All right. So so this is just to remind you. See the orientation? It's complete opposite to this sequence orientation. So the sequence you see here is top strand, and then for this forward primer, because the orientation is opposite, so you need to take the bottom strand sequence. All right. Okay. That's one way to do it. Okay. So one way to do it, it would be. So now we go to the sequence we want. As I mentioned, 2424. Right. We will do 2405 to 2424. So that's 20 nucleotide. So it's here. All right. So the sequence here will be the top strand, but you have to take the bottom strand, so that you're going to copy the sequence. All right. Copy the sequence, and then go to the sequence here. And let's open a new file. And then paste the sequence here, all right? From this AGTTC, you copy and paste here AGTTC, and then you're going to do the anti-parallel, all right? Anti-parallel this sequence. So now this is the anti-parallel sequence. So this will be your forward primer. And then we go to the function. Run a PCR. Run a PCR, okay? And remember this 20 nucleotide. 20 nucleotide align to the target sequence because it's from the PRS 415. So copy and paste. All right, that's layer now. And now we go to the downstream here. All right, downstream here. So let's go back to the downstream. So that means we are going to take the sequence from 413 to 432. All right, 413 to 432, 20 nucleotide. So that's here, and the copy. And since the sequence, all right, the reverse sequence, it's the same orientation to our sequence. So that means we do not need to do the anti-parallel. Okay. So you can just paste here. So that's the forward reverse primer. And now we go to the VMA3. So VMA3, so we can see the sequence is 2483. The length is 2483. If you minus 2000, so that tell you the sequence length is only 483. 483, okay? So now for the forward primer, you need to take the sequence at around 600 to 601 to 640, all right? Because I say 400 base pair upstream of the open reading frame, so 601 to 640, and downstream will be the 1483 plus 400, that's 1883, all right? 1883, we're going to take 40 nucleotide, so that'll be 1844, all right? So we're going to take 1844 to 1883. And now let's go to the 601 to 640, right? So that's a sequence here. So you just copy and paste it here, all right? It's a forward primer. It's here. And then for the reverse primer, okay, reverse primer, it's 1844 to 1883, all right? And then that's the sequence here. And remember, for this VMA3, the forward primer, that's the same direction as the sequence direction, so that you do not need to do the anti-parallel. But for the reverse primer, all right, because you want to amplify, you want to find these complementary flanking sequence for the reverse primer, then you need to copy, and then we open a new file, paste, and then you have to do the anti-parallel, okay? Then we have to do the anti-parallel for this sequence. So now this is the sequence, the reverse primer from the VMA3. We perform the anti-parallel and we copy, and then we paste it here. All right. And before we run the PCR, look at here. We have two different TM. So I also ask you to explain why there are two TM. So the answer is this. Remember that this box, forward primer from this box and the reverse primer from this box, these are the sequence identical to the target sequence. And this box here are from VMA3, which is not identical to the target sequence. Our target sequence is the PRS 415, all right? It's PRS 415, all right? And there's only these two sequences, all right, inside this box, identical to the PRS 415, all right? So that when we perform the PCR reaction, since we use the PRS 415 as the target, so for the first round, for the first few rounds of the PCR reaction, only this sequence and this sequence will be able to adhere to the target sequence PRS 415, and then to perform the synthesis. Therefore, the TM1 60.5, 58.4 indicate the sequence of this box near to the target sequence, which is PRS 415. After you perform the PCR for a few rounds, then you generate a sequence with this completed length, all right, from this 5' to the other end here. Therefore, the second So after they generate these 4 lengths of sequence, then they will be able to anneal 5' from here all the way to the end of reverse primer completely. That's why you have the second TM, all right, the second TM, and also it's a little bit higher than the first TM, all right? So that's why you see 2 different TM. And now you can evaluate the PCR. So the total length is 2092, and then you can run the PCR, and this is a PCR fragment. All right, so this is a PCR fragment, and then you can do the map, the graphic map here. All right, so this is the PCR result, 2092. All right, and they show this red part. This red part are the VMA3 flanking sequence, which is from the upstream 400 base pair to the downstream 400 base pair from the VMA3. All right, so these are the two red sequence, which is not identical to the PRS 415. So that this is the PCR product when we introduce to the cell. These 2 red area will find the VMA3 location and then perform the homologous recombination and replace the chromosome of VMA3, and then put in the insert, which is the LU2, which is leucine. All right, any question about this project? Correct. Yeah, due to that promoter, due to this let me show you this. Yeah, due to this promoter orientation. All right, if the orientation is from here to here, then you do not have to do anti-parallel of this part. You only need to do anti-parallel of this part. So always do the anti-parallel of this part, okay? And then for the target gene, always you have to do the anti-parallel on the downstream reverse primer, okay? So let me show you the VMA3 gene. So the VMA3, so for the VMA3 gene, okay, upstream, the forward primer, you always no need to do anti-parallel. But for the downstream, to do the reverse primer, you have to always do the anti-parallel. And then for the vector, for this target sequence, the template, if the orientation is this direction, then the sequence from here, taken from here, do not need to do anti-parallel. And the sequence you taking from this end always need to do anti-parallel. So that's why in PRS 413, the forward primer is from here, so no need to do the anti-parallel, but the reverse primer from here, you need to do anti-parallel. For the PRS 415, this plasmid, because the forward primer is from here, so you perform the anti-parallel. And then the reverse primer, no need to do anti-parallel. All right? Any other further question?